So therapeutic vaccines represent a viable option for active immunotherapy of cancers that aim to treat late-stage disease by using a patient's own immune system. So what that basically means is that therapeutic vaccines are mainly target to people who have developed cancer or any malignant disease which is already present in a person. So what therapeutic vaccines do is that's their main aim, to target those cells or those malignant tumors. The goal of the therapeutic cancer vaccines is to induce tumor regression, eradicate minimal residual disease, and establish a lasting anti-tumor memory to avoid non-specific or adverse reactions. Again, this line is just stating that that is the main goal of therapeutic vaccines, to just completely eradicate the whole remaining or residual disease of cancer. So if we go on to the next slide, this is just giving more in depth about what the vaccines are and what they represent. So therapeutic vaccination against cancer-associated antigens represent an attractive option for cancer therapy in a view that it has certain advantages, such as it has low toxicity and has a so far excellent profile according to the studies and recent data. So the cancer patients are usually immunosuppressed, and the cancer-associated antigens are self-antigens, which means it comes from the body itself. So therefore, various immune stimulation techniques are again under investigation in an effort to bolster the immune system and to overcome certain tolerance for these self-antigens. So if we go on here again, this is more in detail, just explaining the lines that I mentioned before. The main aim for a vaccine is to stimulate the patient's adaptive immune system against certain specific tumors, antigens to regain control over tumor growth, induce regression of the established malignant cells or tumor cells, and simply eradicate the residual disease. So the basic principles that are needed for successful therapeutic vaccination against tumors include large delivery of large amounts of high quality antigens to the dendritic cells, optimal dendritic cell activation, induction of strong and sustained helper T cells, or cytotoxic T lymphocyte responses. Infiltration of the TME, tumor microenvironment, and durability of the maintenance of the response. So these are the basic principles that we need to keep in mind to aim for successful therapeutic treatment from vaccines. So these steps can be accomplished by several methods, such as reversal of the tumor-induced immune exhaustion by checking the immune checkpoints inhibitors, activation of the dendritic cells, and effector T cells by administering the tumor-associated antigens with adjuvants, or vaccinations with autologous dendritic cells, which can be again loaded with specific tumor antigens. So over here, the adjuvants are basically materials or chemicals which can enhance the vaccine's function by acting or inhibiting certain tumor cells. So there are basically two types of vaccines which we previously discussed, the traditional being the preventive ones, and this is the therapeutic cancer vaccines. So if we go on to the next slide, which is the tumor immunity regulation. How are the tumor cells, how does immunity regulate the tumor cells? So immune cells are driven to the tumor microenvironment via chemokine gradient. So what basically this means is the tumor microenvironment is suggesting that the cells that or the environment that surrounds the tumor cells specifically. So that is basically called a TME. In the TME, the dendritic cells take up the processed tumor antigens and present them on the major histocompatibility cells, class 2 or class 1 molecules, through cross-presentation. So in this cross presentation, what may occur is by two ways. Either it could be the cell's cytosolic pathway or the vacuolar pathway. So in the cytosolic pathway, what happens is that the antigens from the endosomes or the phagosomes are transferred into the cytosol, where the proteasomal are cleaved and then transported back to the ER, which is the endoplasmic reticulum. So everything uh, really happens in the cytosol. So next, the peptides are further edited and loaded with the major histocompatibility class 1 molecules and transferred then to the cell surface. So conversely, after the cytosolic proteolytic cleavage, shortened peptides may be transferred back to phagosomes, loaded on the MHC class 1 molecules, and transported to the cell surface. This is basically the same line explaining it more in depth. So in contrast to the vacuolar pathway, the antigens are processed and loaded onto the MHC class 1 molecules first and the phagosomes or the endosomes instead of going into the cytosol. Antigen-loaded dendritic cells then travel to the secondary lymphoid organs and then which can activate the T cells. So that's where the whole regulation happens in these steps and how it starts, how the immune system then starts to react. So if we go on to the next slide, the neoantigen vaccines are basically act as a platform where the tumor cells or the vaccines we can comprehend the success of the vaccine. So to the large extent, the success of the neoantigen platform depends on the tumor mutational burden. That means that the number of the shared antigen vaccines of any nature per megabase in the tumor tissue. So the more the TMB ratio is for a certain vaccine, that means that there are more chances that the vaccines could be rejected. So if the TMB is less, that means I'm sorry, I did an error over here. It's actually the reverse. With the low TMB, what that means is that the patients with cancer are really poor candidates for that vaccine, which can lead to more rejections. So with shared antigen vaccines, which is different type of platform, there can be many advantages of neoantigen vaccines, but with the shared antigen, what happens is that it's more suitable for the patients who have a low TMB, who have a more suppressed immune system and can't really react in a certain way to the neoantigen vaccine. So this is one main basic advantage over the neo. So the use of this vaccine platforms and appropriate combination therapy may prove shared antigen vaccines to be overall more feasible as compared to neoantigen. We go on to the next slide. These are some target antigens for therapeutic vaccines that I mentioned, of course, taking reference from the paper. As for example, the HER2 neo is for breast cancer overexpression. Human ERT is for multiple cancers, and so on. These are certain more antigens that are expressed for certain cancers, such as melanoma, breast cancer, colorectal cancer. So if we go on further, okay, the efficacy of vaccine. How does that work? So the resistance to clinical active forms of immunotherapy include therapeutic vaccines that may present itself as a direct lack of response to therapy, which is known as primary resistance, or they could develop into following initial responses of tumor to treatment, which is known as the secondary resistance. So these are the two ways or pathways, you could say, that how we can determine the vaccine could have its effect. So many underlying causes may foster this immune escape. These can be grouped into either tumor cell intrinsic mechanisms, determined by the traits of the tumor cell itself, or the tumor extrinsic mechanisms, which can involve the tumor stromal components instead of the cell itself. It's very important to realize that the same factors determining the initial resistance to immunotherapy may also drive the occurrence of secondary resistance. So if we go on to the next slide, this is the direct administration of vaccine. These are certain routes by which the antigen vaccine can be injected into the body. So they can be various routes such as intravenous, intramuscular, subcutaneous, intracutaneous, which introduces the antigen into the antigen processing pathways of dendritic cells and the vaccine draining lymph nodes. So there are certain factors which determine the success of the vaccines that are being injected, which some of them are as follows. The number one being the type of the antigen, such as MHC1 or MHC if it's MHC1 or MHC2 restricted. The second being the antigen dosing, how much antigen is being given. Third, the adjuvant that is being used. And number four, the administration route. So far, the satisfactory sources of the tumor antigen delivery have been included by DNA, RNA, and synthetic long peptides. So if we go on to the next slide, this is just briefing a little bit about DNA vaccines. DNA vaccines are easy to manufacture, carry built-in adjuvants, and like RNA and peptide vaccines, they represent a concentrated form of tumor-associated antigens, but require extra steps of transcription and translation before cross-presentation by the dendritic cells 5 and 112. So these vaccines are most effective in driving sufficient antigen processing and presentation for the induction of T cells, helper T cells, and killer T cells responses when administered at really high doses via IM route. So if we go on to the RNA vaccines, they differ from DNA vaccines by being they are really straightforward, but unlike DNA vaccines, they don't need the ability for transcription and are more closer to the protein antigen expression. So in that way, they are more straightforward and presenting or expressing themselves on the MHC molecules. So the synthetic long peptide vaccines are often consisted of the MHC1 binding short peptides. So these vaccines generate robust T cell responses when they were used with a combination of, again, an adjuvant and or a monotenide on toll-like receptor 9 ligand. So these vaccines basically were more effective when they were used with the adjuvant. So the mechanisms that compromise the vaccine efficacy. The resistance to clinically active forms of immunotherapy, including therapeutic vaccines, may present itself as a direct-acting response to therapy, which we discussed previously, known as the primary resistance, or may develop following initial response of tumors to treatment, which is known as the secondary resistance. So there are many underlying causes that can foster, again, we're just explaining that. So these can be grouped either into intrinsic mechanisms or extrinsic mechanisms. So if we talk about advanced tumors, which are more generally resistant to the ICI, we can have multiple phasic approach instead of being a direct one. So in those cases, what happens is that usually there's a first phase, which involves the tumor debulking with the surgery, chemotherapy, or radiotherapy, which can also reduce the immunosuppressive pathways. After doing that, there could be a second phase, which would involve the vaccination and help reduce the remaining residual disease and also establish an immunological memory so that it could prevent the body from remission and having basically a memory. So the vaccine could be based on viral antigens, shared tumor antigens, or personalized neoantigens administered as peptides, RNA, or DNA, which we previously discussed. If we go on to the conclusion, this is the last slide. So in the conclusion is that immunotherapy has offered definitely patients with cancer new hope, but unfortunately it's not effective for everyone for all the cases, despite having a very good potential of cancer vaccines for tumor immunotherapeutics and so many evidence that cancer vaccines are able to activate the immune system and generate a response by acting on the tumor cells. But again, this could not be for everybody because as everyone has a different type of host, and there could be many reasons where it could not elicit a same response for everyone. So cancer vaccines, however, do offer an attractive approach to synergize with currently available immunotherapeutic strategies such as the immune checkpoint inhibitors, ICI, or the oncolytic viruses, or any other immune modulators such as the several cytokines to boost the anti-tumor immunity. So this approach definitely offers many clinical benefits